changes the color, but you go to see the sunrise, so the sun didn't rise yet, unless you were too late. Right? So what do you look for? What do you look for? What would you look for? Ah, yes, the horizon, right? So suppose that you were stuck in an island in the Pacific Ocean. How would you know the horizon? Which direction of the horizon to look for? Somebody has said it already. You see the brightness of the light, perhaps? Okay, so you know. And then you wait, and then what happens? The sun rises. So you know what's to expect. The problem in science is that we are forever looking for the horizon. We have this ocean of knowledge and understanding, perhaps, but the horizon is forever moving. And we don't know exactly what's on the other side. And that's the problem. And that's what we try to get our students to recognize in terms of what's going to happen in the future. So what is synthetic biology? And what is synthetic biology? I don't know what it is. And the reason I don't know what it is because it's about young people. It's about Kiana and Anna and Travis and people of that age. We old fogies don't really understand what synthetic biology is. So I'll stumble along and try to give you some ideas of what I think it is. The problem, the need of what can be synthetic biology is that genetic engineering, which Ken said, and the previous speaker also, Kiana's friend, said, started around 1974. Genetic engineering, how to modify living organisms to do something like make insulin or modify plants to make better food or something like that, have been very, very slow. And people didn't know, although they knew how to do it to some extent. Every problem is unique. And so we needed PhDs and postdoc experiences to come to the lab and spend a lot of time. And you need to pay these people, so it's expensive. And the components, that is, the things that people use pipettes for to transfer from one tube to another, those things cost a lot of money. And they're not reconfigurable, so they're dedicated to a specific task, and that's it only. And if you need to change something, then you need to spend months trying to develop that, and then put another component in. And that adds to the cost. The technology of genetic engineering is rather inflexible, slow, and biologists love tinkering. PhD biologists like me love tinkering, like tinkering in one's garage. They don't know exactly what's going to happen. In order to try to understand something, they shoot the car with a machine gun, like what Ken was describing. You shoot the plants or the organisms with mutagens, chemicals, radiation, and cause mutations, and then figure out, what is broken. And then from what is broken, try to figure out what the parts are. Okay, so uncertainty is part of the problem. So the difficulty and uncertainty of various acts in genetic engineering caused, just giving up. It's too difficult. So most investors don't like this. Who likes? You remember what's the expected utility formula was for stock? Okay, so if you plug in, you'll see that most things in genetic engineering they're rather low expected utility. And so from an investor's perspective, it's $15 to check in the bag and $25 if you want to arrive at the right destination. Most of the time it's misplaced, and most of the times you don't get your bag, and you have to pay for it anyway. So that's the problem with genetic engineering. So this is a huge definition, and I don't pretend to put them together, but synthetic biology is somewhere in the overlap regions of these three balloons. One, the yellow, is modular, reconfigurable components of specified biological properties. Something that looks like modular. That's the keyword, modular. And reconfigurable. So you can change this a little bit by simply combining modules. Then molecular, the blue balloon. Molecular devices for reporting, editing, controlling, mimicking cellular function. You've already seen that. Kiana's CRISPR immobilized on chip is a device for reporting a molecular signal, something like that. And then something that we really haven't talked about very much here, and this is a large part of our instructions to our MBA students and various other students in KGI, is this genome scale engineering of artificial organisms and dedicated to a specific function. And synthetic biology combines these three balloons somewhere in the middle. So in 2018, in the first quarter, synthetic biology companies raised about $650 million. That's a record for synthetic biology, for anything in biotechnology. And it's thought that it is going to be a $2.5 billion industry very soon. So we are looking at the future, at the horizon, beyond the horizon. And synthetic biology application is not just in one field. You can't read this bottom, but these are different company names which do many different things. Only very small section of that is pharmaceuticals or drug delivery, or even have anything to do with the health. So there is a huge potential, stars, whatever, below the horizon, which we can't see it yet, that has nothing to do with drug delivery or human health, but maybe many other things, and I will talk a little bit of that. Ken has talked a little bit of that in terms of food production, but I'll elaborate a little bit more. Now, synthetic biology is really not biology. This you need to understand. It's not really about biology. It's about engineering with biological materials. And the engineering is more like electrical engineering and less like bioengineering. And from Kiana's talk, you already know that behind this, there is a lot of electrical engineering, all of that chip, and field effect transistor. And she briefly mentioned carbon nanotube as a transistor. Those things combine together to create a discipline of synthetic biology in which biological molecules enable novel kinds of engineering which are more similar to electrical engineering. Now, if you go go to this site. I hope you can see that. No, you can't. How do you get the image?
I think you have to drag it off the screen.
I see. All right. I think this is going to waste more time, so I'm just going to go back to this. I'll make the slides available and you can see the video on the back. If you click that, you will see somebody called Drew Endy, who is sort of the evangelist of synthetic biology. He's a professor in Stanford. Started this entire field by coining the term synthetic biology. He was trained as a civil engineer and then came to do a PhD in biology. And very quickly he found out that biology is very messy and nobody really knows how to make things. Whereas civil engineers know exactly how to make things. Like electrical engineers of those days or civil engineers of those days. There used to be a big, fat book of modular components that you find out, okay, this is a transistor of such and such characteristics, this is a rheostat of certain such characteristics. You put them together and you make a device. There is no such thing in biology like that. So he wanted to transform this entire field such that there are these modular plug-and-play devices in biology that you can Google and get them and put them together and make new biological devices. And so he wanted to engage the very properties of the biological uncertainty, the messiness that we're talking about, and the variability that is caused because of evolution and things like that, and use those to empower the engineering principles. And he also wanted to recast the genetic tinkering as a real engineering discipline. So he started that back in 2002, and since then, a lot has happened. So, to summarize, it is an engineering discipline. It uses engineering concepts, especially those of electrical engineering, to design biological organisms, which never existed before. It very rapidly fabricates new pre-designed meaning in your head, you know the design because you have using a computer-aided design principles, you have figured out exactly what to do, so that's pre-designed, so you want to make it very quickly in material terms, and then assemble together the pre-designed modules to enable new biological function. And I'll give you a cute example. Let's say, do you recognize the profile? Who it is? Alfred Hitchcock. So,
I think you need to back up a yeah.
on a petri dish, you see the image of Alfred Hitchcock projected. Okay, so this is light, this is dark. So you create a little cutout of Alfred Hitchcock, and you project this image on a petri dish. And lo and behold, bacteria that are spread on the petri dish finds the edge and swims and accumulates just at the border. Such bacteria didn't exist in the world. They simply made it. Very quickly, they made it. And they made it with the help of a few high school students who came up with the idea using a very simple logical rule. And the logic operation was X and not Y is 1. In other words, the gene is 1. And the gene causes the bacteria to swim. And the conditions are X is light, presence of light. And Y is, or the negation of Y, is in the vicinity of cells in no light. So when you put the two logical operations together by an AND gate, the gene turns on and the bacteria swim. And the circuit is like an electronic circuit, which is shown on the top. You can't see it, you don't need to see it, but it looks like an electronic circuit that actually recapitulates this logical operation. And when it does so, bacteria just swims and arranges itself at the borderline of Alfred Hitchcock's profile. Such an organism obviously didn't exist, but they made it with the help of a few little tricks. So from such cute device which has no practical utility, what can you do? Well, why not design a safe synthetic delivery of bacteria that is totally safe for human beings if such a thing exists? An agent that resembles a living organism, for example, bacterial cell, to detect the edge between the tumor and non-tumorous tissue in the pancreatic islets, where pancreatic tumors arise, then release the drug instead of swimming, just going there and sitting there. You design the bacteria such that you release a drug from the bacteria to kill the tumor cells at the edge. And the logic operation will be tumor markers. Are there tumor markers, meaning are there gene signatures, gene expression signatures that resemble a tumor and can be distinguished from a tumor from a normal cell and in the vicinity of normal islet cells, so giving rise to pancreas-specific tumor recognition. And then turns on the gene, release the tumor cell-specific poison, which is going to kill the tumor cells. Well, that looks like a science fiction, but actually it's not. It has already been done. Way back in 2006, 3 people in UC Berkeley had already done that and shown that it actually did work in mouse. From there, going into human beings is a big leap because you don't want to inject living bacteria, such as E. coli, which was used in the mice, injected into a cancer patient, because who knows what the E. coli is going to do. In fact, it's probably going to kill the human being by eliciting immune response, massive immune response in the patient. So you don't want to do that. You want to do something else. And one of the things that you want to do is create cells that don't exist in the natural world and therefore can be completely safe, made to be completely safe for humans. And a little step on that was taken in 2010 by Craig Venter, who has a company, what is the company called? Synthetic Genomics, Inc., in San Diego, La Jolla. And that company, along with Craig Venter's nonprofit institution, create the first artificial organism in which every gene was chemically synthesized. So you could now chemically synthesize an organism which would do this function, which may be designed by altering its totally synthetic genome to be completely harmless to human beings. It hasn't yet been done, but people are trying to do that. Now, is it all going to be reality? One of you, I forget who, sent me a invitation for registering to this conference on the 26th, Friday the 26th of this month, Biotechnology 2.0 in Amgen. So Amgen is thinking about it because in the program at 1 p.m. there is Mike Elowitz of Caltech, and at 1:30 p.m. Alex Marson of UCSF. Both of them are synthetic biologists who are talking about redesigning protein circuits within organisms, and Amgen is very interested in learning how to do that and what the implications are going to be for Amgen's drug development strategy. So we are not that far away from applications to human health. But there, of course, there is more to biotechnology than curing diseases. So let me ask. Let's see. Yes. What do you think is a lifetime risk for dying of car crash in the U.S.? Anybody has any idea? What's the risk of dying of car crash? You can be injured but not die, but not dead. But what's the lifetime risk? You could Google it and you'll see it's about 1 in 572 or something like that. 1 in 500, 1 in 600. Lifetime risk of every individual who live in the US of dying in car crash. What's the lifetime risk for a person born in US of dying of cancer by the age of 80? Anybody guess?
1 in 20?
1 in 20, very good. Exactly, 1 in 20. How about dying of heart attack by the age of 80? Lifetime risk. 1 in 15, pretty good. It varies from 1 in 5 approximately to about 1 in 30. And depending on no risk factor, 1 in 5. Not 1 in 5, 5%. 5% chance. To 1 in 30, to 30% chance if you have at least 2 risk factors. So it varies from 5% to 30%, you know, you're pretty close. Those of you, none of you, were born or lived in the 1960s and 70s, but I did. And at that time, it was calculated that the lifetime risk of dying of nuclear attack was about 80% estimated. Well, we sort of think that that may not be true anymore, but who knows? We don't know that. What's the lifetime risk of dying from climate change issues? Very difficult to calculate. But there are some estimates that have been made. Climate change is simply not just increasing the temperature of the earth and you feel hot or warm. It's doing all kinds of things, such as increasing pestilence. In other words, malaria, dengue, and some parasitic diseases in countries where such diseases don't exist. It's increasing due to glacial melt and polar ice cap melt, increasing local sea level, ocean level, and as a result endangering lives of people. For example, I don't know whether you can see this. Yeah. I'm going to say it because I don't know how to manipulate this anymore. But if you look at this month's, that is July of this year, National Geographic, you'll see a beautiful article where a friend of mine actually has taken all the pictures. And you'll see some fantastic pictures, beautiful pictures of very sad events in which people are losing their home. And people are losing their home in the deltas, low-lying areas of Southeast Asia, where glacial melt and therefore rising local sea level is inundating the islands and the deltas. And as a result, they're becoming destitute. Their livelihood is being destroyed, their homes are being destroyed. For your information, I was born about here, not too far, and my family still lives there. Part of my family still lives there, not too far. So you think that you're very far away, but I can't think that. Estimates of additional deaths due to climate change in 2030 to 2050 under the current trajectory, under the current rate of global warming, is about 529,000 additional deaths per year from the British medical magazine Lancet. More than 100 million people will be forced into extreme poverty by 2030. That's an estimate of the World Bank in 2017. About 3.2% reduction per person of food availability globally averaged. These are huge problems. What are the land areas that are to be affected by climate change? These are color-coded. The dark brown areas are least affected, and you can see that North America and Europe are least affected, whereas places like India, well, most of Africa and Southeast Asia are very, very acutely affected. And the numbers in the table are so-called DALI, or Disability Adjusted Life Year, is a measure of the overall disease burden because of certain factors. And global and climate change is going to affect the overall health of populations, and those numbers are in the table. Using those numbers, you can calculate very simply the economic impact merely because of loss of human life or disability in response to climate change. And that's about $480 million lost in the economy per year in a developed world. Nothing much compared to the GDP of the developed world. But in the developing world, it is actually quite perceptible, about $21 billion, simply because of diseases and lives lost. And these don't count the values of the resources, economic resources lost due to disaster relief, war, or political and social upheavals that are predicted to occur because of this. So although we think that we can build a wall and insulate ourselves from the countries in southern hemisphere or most of Africa and Southern Asia is going to come back to us. So what are we going to do about that? And we can do a lot. We can be less carbon dependent. In other words, produce things like gasoline, perhaps, using microbial fermentations, using, in other words, renewable energy. And how do we do that? Well, this is how we do it, using synthetic biology. So synthetic biology has already been engaged by various laboratories, various institutes, and companies already. ExxonMobil is heavily investing into it also. They are redesigning microbes to generate gasoline out of cellulose pulp from grasses and things like that. And these reagents, the genetic reagents they're designing, they're all by synthetic biology. And I'm going to skip this. Just to recapitulate, the definition of synthetic biology is modular reconfigurable biological manufacturing unit. So the same things that can produce gasoline can be used to produce small molecule drugs. Those links, and I'll make the slides available, those links, if you click on, they're going to take you to synthetic biology open ware system in which people are trying to make it available to everybody, to people who tinker things in the garage, so that we hope that just like software industry has been dependent on people, ordinary single individuals that are tinkering with producing openware software. People can make openware biological entities by tinkering in garages, and therefore those links, if you click on them, they'll take you to places where such resources are present, and you can simply order by online order little pieces of DNA, and all the methods are present, and you can put them together, and these are modular designs, and you can put them together to make novel devices that can make wonderful things. There are worldwide competitions called iGen, or International Genetically Engineered Machines. Every year that happens in Boston, used to be at MIT, but now it's outside MIT because it's a huge competition. Every year, high school and first and second year college students from throughout the world come and make biologically engineered machines, costing approximately $2,000 to $3,000, and they get the money, raise the funds somehow, and come and compete, just like in a robotic competition. They make biologically engineered machines that do this or that, like the dark light edge detection machine that you saw, but more interesting stuff. So the freeware concept is that it levels the playing field. Instead of large companies like Monsanto, for example, because of their resource-rich capabilities, coming up with genetically altered organisms to do good things and then be blamed for things that they probably should not be blamed for. Now, the onus is on individual entrepreneurs who, with much less resource, are enabled to do these things. The problem, of course, that comes with it is how to protect the intellectual property so that they can make businesses viable. The concept is no different from software manufacturers that make software, and sell them at very low price and have copyrights. So that issue is going to be solved eventually. And the old ways of negotiating the IP landscape is going to fade away, most likely in this field. So synthetic biology is a model for accelerating biotechnology. You have a product concept from where you have a conceptual construction of genetic elements and then making those very cheaply. You integrate them into cellular systems and test and optimize, and you then make a product And in between, you have all kinds of computer modeling and things like that, which also, the resources for which also are available totally free. Here is an example in which a medical application for the developing world was made. A small molecular drug against malaria, artemisinin. Artemisinin is one of the last frontiers of small molecule against malaria. Malarial parasites throughout the world are becoming resistant to most anti-malarial small molecules. Artemisinin is one of the few that, if I were speaking last year, would have said, so far no resistant malarial parasite exists against artemisinin, but unfortunately, few now has surfaced. But back in 2006, artemisinin, which was available only from a specific plant that grew only in northern Vietnam and China border, instead could be synthesized in yeast at a very low cost. 500-fold increase was obtained by using synthetic genetic principles, and 10 million-fold increase was achieved in E. coli using similar principles. And so there are now companies that have acquired the license and are making synthetic biology-made artemisinin and marketing in the developing world. Rapid engineering of mammalian cells with modular RNA scaffolding is a health-related problem, and that RNA scaffolding directly leads to CRISPR-mediated control of gene regulation very similar principles that Kiana was talking about. Kiana was talking about detection, but in this case, we are trying to re-engineer mammalian cells, perhaps human antibody-producing cells, to direct them towards a cancer cell by altering the regulatory circuits of the cells using tailor-made CRISPR-Cas9 products and targeting them to specific gene locations in human cells and letting them back into the human patients to kill the cancer cells without giving rise to enormous hypersensitive reactions, which is a problem with the CAR-T technology. There is another one. For example, back in 2014, they went from stem cells to neural cells within 4 days. Normally, it took about 4 months, but re-engineering human-induced pluripotent stem cells to bipolar neurons. Bipolar neurons means there's a region in the brain which has this kind of neurons in just 4 days by using synthetic biology. So these are some of the potentials, and so we have potential for novel biosensor devices. Again, I can't click that, but if you did that, you would go to a company website called Pairwise Plans Inc. If you Google pairwise.com, you'll go to that same site, and it tells you that it's a new company that came into being only a year ago, not 2 years ago, 2017, and is currently raising funds to genetically engineer food plants, food crops, plants of all kinds. And it is in its scientific board of directors and founders are the people who invented the CRISPR-Cas9 technology and the leading synthetic biologists of the world from MIT and Harvard. And they're going to take 100 new people within the next 1 year. And I hope that some KGI graduates ultimately find employment in such companies, ultimately. There are other factors involved with synthetic biology. For example, the safety features. As I told you, you don't want to inject a patient with E. coli with altered circuits. So the organisms would have to be safe. or you don't want to let genetically engineered organisms that compete with other organisms in the ecosystem to reduce the carbon dioxide load of the atmosphere, for example, which is one area where DARPA is actually interested in and is funding a lot of people to do that. But you don't want to put in artificially synthesized organisms that compete with other organisms and disturb the ecosystem. So you want to produce safe organisms. So there is a lot of work going on which has multiple safety features that can be turned on and make the organism suicide if something goes wrong. I'm going to skip this and just show you from December 2010 the report of the Presidential Commission for the Study of Bioethical Issues in Synthetic Biology. So there are guidelines, provided 18 recommendations, and all synthetic biology practitioners would have to abide by these 18 guideline recommendations in this country, and this is being adopted internationally as well. I think I'm going to stop there and provide the summary and the horizon. Of course, that horizon is not the earthly horizon, that's the lunar horizon, and the one that is rising is the Earth and not the Moon.